did before. We won't use the server today. We will just go on the web and do some manual work on try to find out protein structure and function. The reason why we will do this is that in many cases you are ending up with a list of genes or proteins at the end of your experiment you are interested in. And let me just click from my email. And many times there are unknown or hypothetical proteins among these genes or proteins. And if it's not that much, if you are, especially interested in that one genome or organism, and you really want to know as much as you can from that gene, then it worth the time to go after each one of them manually. So the main reason why, and let me just share my screen instead of. So I will just put the blast here while I'm telling these things. So there are many reasons why you will end up with this so-called hypothetical protein. So just imagine that you are using, for example, proca. We are remaining in the prokaryotic world today, but this is actually quite easy to apply all of these things what we will learn today for eukaryotic genes and proteins as well. Actually, there are much more tools, to be honest, for eukaryotic proteins, but we will remain with the prokaryotic genes and proteins today. So you have your new genome sequence. You are annotating with proca, and then you just see quite a lot of records with the so-called hypothetical protein. I will show you how it looks like, because thanks to Ibrahim, I have a few. The reason why, so it looks like this: conserved hypothetical protein, or a protein fragment, or hypoprotein putative, but nothing else. So not really too much about the function, or no gene names. So in this case, I don't want to tell too much about this project because it's not published yet. And of course, this will go out the YouTube as well. But it was a TRADIS experiment for Ibrahim, and he obviously got genes that were differently destroyed or knocked out in different environmental conditions. So what you think after a TRADIS experiment, I don't know how much of you doesn't know or don't know what's the TRADIS, but it's actually a very easy tool to randomly knock out genes in prokaryotic genomes by inserting a transposon, and you can extract variable information, for example, the essential genes in genomes, or essential genes in many different specific environments for that organism. So of course, Ibrahim ended up with a list with some known genes and with some unknown genes, and we will help him today to try to identify these proteins, or try to give a bit more information that just what proca gives as protein fragment or hypothetical protein. But this should work for all of you if you want to identify or know a bit more about your proteins. There will be a few good things. I already checked these proteins, and I think we will be able to give some extra information. But there will also be a few ones where we won't be smarter than before. So the basic tools we will use, and we don't really need this anymore. So we will use, of course, the BLAST, the protein BLAST today. So the NCBI protein BLAST website, we will use the EMBOSS PEP info. So if you just Google EMBOSS PEP info, then this will be the very first link we will use the UniProt, which is one of the best protein database. It's not just very up to date always, but it's also very good, very well annotated. So in many cases, you will find a lot of very useful information about proteins in UniProt, way much easier and much more than the NCBI site. We will also use the PFEM website, which is the website for identifying domains. And we will also use the IUPRED, which is one of my favorite programs. It is actually just a protein disorder predictor, but as you will see, it will give us an interesting first look for proteins, which can be used for quite a lot of things actually. Even for predicting PFEM domains, sometimes I'm just using the IUPRED because it's also predicting PFEM domains together with the disorder prediction. The reason why, yeah, I think I started saying that I didn't finish this. The reason why Proka or any other annotation program will give you a non-definitive product name or gene name or protein name is that, of course, they are not specific for specific organisms. So if you are interested in E. coli, then you have a much higher chance to get back more definitive gene or protein names than if you are interested in something which is like a soil commensal bacteria. In this case, this is a Staph aureus, so it's not a less known, it's a very well-known organism actually. But even in Staph aureus, there are so many different strains, there are so many different genes that we don't know exactly about. And also, even if we know quite a lot about Staph aureus, it's not all of these informations are included in these annotator programs, simply because it would be too much. So if you think about the process, what we did, for example, before when we annotated the whole genomes, so you have to wait minutes, several minutes per sample. So if you want to include all the information we have in all the databases, probably annotation of just one single isolate genome would take hours. So there has to be a balance between the amount of information it can hold and the amount of information or the amount of time the process is spent. Of course, if you are specifically interested in one or the other species or organism, then you can actually make some extra databases for the Proka as well. So if you know that your next 2 years work will be about Strep suis or Staph aureus or just E. coli or Campi, then you can include much more information about these species. You can download a lot of different whole genome annotations from the NCBI, for example, not just including one reference genome, but including several reference genomes, several annotated reference genomes, and you will have a much higher chance to identify proteins or genes with more definitive names or descriptions. So I will copy the protein sequence from here to the chat window. So if you want to do the same thing as I'm doing, just please follow me, especially if I will find the chat window. Again, please handle these sequences with, oh my God. Let me just copy this again because this looks awful. I will delete the spaces and new lines from here, and then it will be a bit, oops, I think I deleted an amino acid. Yes. You don't really have to delete spaces and new lines. Otherwise, all of these programs we are using, they just simply ignore white spaces. This will be much better now, I think. Oh yeah, so this is much easier to copy from the chat window. Okay, so if we are here in the IUPRED, this is IUPRED 2a. So if you Google for IUPRED 2a, this will come up as well as one of the early hits, I hope. But it's an iupred.egu, I think. I don't really see the address. So if you just simply click on submit, then you will see something like this. So don't really think too much about the blue line at the moment. The red line is much more important. And also this one down here, the PFM line, is important. So the red line is showing us the disordered tendency of this protein. This is clearly not a disordered protein. The disordered protein means that the protein doesn't have a well-defined three-dimensional structure. So it's like a wibbly-wobbly linker region or something like that. If it would be a disordered protein, then you would see the red line well above this 0.5 line. But this is not all what we can read from this diagram. There is at least one more thing. So it's also quite good to see that we have the red line well below the 0.5 line, which means that we probably have a globular protein here. So we have a protein which has a structure. And also there is something here that is a bit strange as well. So we have about 20, 30 amino acids that seems very hydrophobic. We will see way much more hydrophobic proteins today. But even this one, if you see something like this, is there anyone who has an idea what a 20, 30 amino acid long, very hydrophobic region means? It's usually very, very well defined in size as well. So 20, 25, 30 amino acids as a length and extremely hydrophobic. If there is no idea, and this is actually
inserted in the membrane.
Sorry?
Inserted in a membrane.
Yeah. So this is usually the length of one alpha helix that is going through a membrane. In most of the transmembrane proteins are alpha helical. There are some beta barrel transmembrane proteins, but they are very special and not really common. They can only be found in bacteria, actually. There is no beta barrel proteins in eukaryotes. Actually, there are, but I think they are only in the mitochondria, which we can, of course, debate if it's a eukaryotic or an old prokaryotic organism inside the eukaryotic cell. But if we are just looking at the most common way how a protein is going through the membrane, it's usually one alpha helix which is going through, and the length of this alpha helix is usually about 20, 25 or 30 amino acids. It also depends on the orientation of the helix which is going through. So there is a very good chance that this one is a transmembrane region, which is again something that's already an information for us. That means that, okay, we don't know the protein, we don't know the function, but there's a chance that it's actually a transmembrane protein. The other one, which is very important here, that we already have a known domain, which is called thioredoxin 5, and you can also click on this. It will take you to the PFM website, where you can see that this is actually thioredoxin 5. Yes, so this is the name of the domain, and that we don't know anything about it, which is not very useful. But the reason why this is thioredoxin 5 is probably because it is very similar to other thioredoxin domains. But it's maybe a little bit different in some structural or sequential aspects. But if it's very similar, even that similar that the name is the same as other ones, then you can always just go back to these ones, as you can see here, that are very similar in some ways to this one, and probably you will have much more information on these links. So if you just click on the thyrodoxine, which is probably the oldest domain in this family, in the BFAM, and you will see way much more. And you can see that proteins that have this domain are usually play a very important role in biological processes, but this is not extremely important. But I think what is much more important that, for example, these domains are very often involved in response to reactive oxygen species. So for me, that means that this protein is probably like a stress response or some kind of defense mechanism for the bacteria. You can, of course, from here you can go quite a lot of different ways. You can read a lot about the protein. You can also usually have quite a lot of citations here as well. So here are the links. Oh yeah, the references are here. So you will also, there is also a good chance that you will find a lot of prokaryotic links about these proteins. So as soon as you know something like this, it's actually a very good answer to the main function of the protein. You can also, of course, blast the protein, and we can do it for this sequence. Just use the default settings of the blast. You don't have to do, and this is the blast P, of course, because we are blasting protein. And of course, it will give us the one of these. It's an already known Staph aureus gene, so it's not a novel gene, but I think it will also give us some not very descriptive results. So I think this is the point where for one protein, this stage ended, and I don't really want to go further, because from here it is all about finding literature references, finding more information about this specific domain function. As you can see, this domain is almost the whole protein. Of course, you can think about what we have here and what we have here. I mean, this is very, if this is a transmembrane protein, I don't really know if the thyrodoxine domain can be in the membrane as well. The BFAM actually, so if we looked at the BFAM homepage and we are doing a search here, here you can upload files, but if you click on this HMMR website, I will open in a new tab. Here you can also use just a simple row sequence. And what you have to do is to be sure that at least the BFAM is here, and also be sure that the gathering threshold is used. This is what we already spoke about last time when we spoke about the HMM-based sequence search or BFAM search. This is the threshold which is very useful to avoid false positives. And you just simply submit. And as you can see, it's a domain as well. It doesn't say that there is a transmembrane region there, so probably it's just a part of the domain. But it's also interesting that, as you can see here, it has a kind of strange C-terminal. So it's not really a complete one, which means that it's probably not a full domain. But as far as I see, So again, if you see something like this, then it may be interesting to see how much shorter the region is than a normal thyroreducin domain. Okay, it wasn't here. And I will just go here. So you can always look at the alignments or actually the HMM logo and see the size. It's 103 here. I don't think it's way much more shorter than a normal thyrodoxine. Let's see the 5. Yeah, so this one is 186 amino acids usually, and what we have here is 160. So it's about 20 amino acids shorter, which is not really significant because we have most of the domain present. Okay, let's go for the next protein. So I think we are happy with this one. I don't know if Ibrahim, you are happy with the answer of the protein, and
yeah,
I think it's much easier to go forward from here. If you need like functional annotation, then you can find, I'm pretty sure you can find prokaryotic genes with the same domain. I think it's also on the PFAM side. So if here you can see the species, actually it is quite often in prokaryotes. So maybe you can even find here yours and see if there is any protein with a more annotation, or if not here, then you can just go back to one of the other family members of thyroidoxine, and then you can find proteins that are a bit more functionally annotated.
Okay.
Right? Let's go for the next one, just to have something with a bit less success. Right. I will use again this window just to clear up the spaces and new lines, and we'll copy and paste the sequence for you. Let me just okay. So if we look at the IUPRED prediction for this one, it is extremely unusual to say. So what you see here is actually the blue and the red line also. I mean, as I said, the red line is, for us, is the more important now. They are sitting extremely on the x-axis. And if you look at the sequence, but even if you don't look at the sequence because you don't know from head which letter means what, let's just put this into the PEB info. So this should be open as well. Just copy here the sequence and say submit. And the thing that has to be seen very, very quickly, what we also see on the IUPRED prediction, is that this protein is extremely hydrophobic. So what you see here, these are the non-polar residues. I don't know if there is any I mean, here is what you can see, the charge residues. There is like almost no charge residues and only a few polar residues. Everything which is non-polar is hydrophobic. So this is extremely hydrophobic. To be honest, I'm not completely sure that this is a real protein. For me, it's a little bit too hydrophobic, to be honest. But if you go for this HMML website and go back to search and paste the sequence here, then it will actually Oh no, it doesn't find. Yeah, it finds it. Yeah, it finds 4 uh, potential transmembrane region, which is not because the whole thing is extremely hydrophobic. But at this point, I have to tell you something which is, I think, extremely important to know. If you know that a protein is a transmembrane protein, for example, you are looking at the expression of a protein and you see that the localization is in the membrane, or if you make a membrane prep from a cell and you will see your protein remaining in the membrane, then you can use different predictors to predict the transmembrane regions. So as soon as you know from somewhere else that the protein is a transmembrane protein, the predictors are quite reliable. Okay, let's say they are very reliable to predict which regions are the transmembrane regions. But if you don't know if a protein is a transmembrane or not, then this is not extremely reliable. I'm pretty sure that if I just copy and paste here a very well-known globular protein with a big hydrophobic core, there is a chance that a transmembrane predictor would predict a transmembrane region there. So, oh, that's okay. This one is also doing some blasters actually. So we can see that this is already known in the UniProt. This is the UniProt ID. So it's a very useful link here. The E value is extremely low. So probably this is a very good alignment. As you can see, if the middle line is a letter, that means that there is a complete match between them. So as you can see, this is already a known protein, but known as an uncharacterized protein. So we are not much better with this. We are not even much better if you try to click on this link and you just go to the UniProt website and you don't know anything else about it. It is, again, say that there are 4 transmembrane regions, but again, this is just a prediction. So you have to be you have to handle this with care. The problem here, so we don't know anything about the domain. We don't have any known homologies here. So as you can see, everything that is a little bit similar to this protein are all uncharacterized proteins. Even if we believe that this is a transmembrane protein, that doesn't have too much, because the functional annotation of transmembrane proteins are way much worse than the globular proteins. So, so from 2 proteins, we were able to identify one, but we won't be able to identify this one. I don't think we will be able to say anything else than it is potentially a membrane protein, but we have no idea what's the function. It's extremely hard to find out a function for transmembrane proteins if you don't know, if you don't have any homology to something else. All right. Let's go for the third one. Sorry that I wasn't more helpful this time. Also, if you have longer and shorter proteins, you have much higher chance to know something or to find out something about longer proteins. The shorter ones are not extremely informative many times. Okay, let's just do the search here for this one first, and also, okay, I will also copy and paste the sequence. The protein free there, but I hope you can see that. So I will submit it here, and I will also submit on the iPad website, and then both servers are working for us. Again, this is extremely hydrophobic, so there is a good chance that it will be annotated as a transmembrane protein. Yeah, so we have one transmembrane domain here, but not much more. I think, as far as I remember, this is the other one which is not extremely informative for us. But we can see, we can wait a little bit here. The problem with the transmembrane proteins is not just that you can, it's very hard to predict anything bioinformatically, but it's also way much harder to work with them. So, you know, you cannot really express this protein in other cells because you don't know if it will go into the membrane or not. If it's not going into the membrane, then it will just aggregate inside the cell because it's too hydrophobic. So that's actually quite a bad news if you are highly convinced that one of your best hit comes out as an uncharacterized transmembrane protein, then it's not really a good news for your future work. I think it's a bit better to try to avoid to know about to work with try to work with these proteins. Of course, you can. I mean, I can imagine that one of the next step for Ibrahim is to do targeted mutagenesis. So if you think that this protein, if you continuously think that these proteins are, or especially these two small potential transmembrane proteins are really important for that specific function you are looking for, then you can check again by doing mutagenesis and knock out these genes, not with TREDIS, but specifically on a strain. And if you see the same phenotype change, then maybe it's worth to go after it. It's also true that if you have a transmembrane protein, in terms of, for example, the immune system, I mean the host immune system, they are accessible. So characterizing them functionally or expressing them or especially crystallizing them is way much harder. But maybe in terms of target, even if we are thinking about vaccine or if we are thinking about some kind of antibacterial therapy, maybe they are a very good target proteins. So at least something you can which can cheer you up if you have to work with transmembrane proteins. For me, this is just loading and loading and not really giving me any results here. So I will also try to search in NCBI. As far as I remember, there was some kind of lipoprotein, I think here as well. It was annotated as a lipoprotein, which is, again, not really definitive. So, you know, in eukaryotes or in a human body, we call those proteins lipoproteins that are usually together with lipids, like the proteins that are involved in the formation of micelles. So like the HDL or the LDL, transporting lipids in the blood. But again, it can be just simply the reason why they wrote lipoprotein here, or why these proteins were first annotated as lipoproteins, because they are extremely hydrophobic, and of course they will always be together with some kind of lipids, because lipids are hydrophobic. So again, this is again something you have to be very careful if if something like this is written here, that doesn't mean that it's actually a traditional or a real lipoprotein. Maybe it's just a membrane protein, which is, I think, much more should be called as membrane or transmembrane protein much more. Yeah, they are extremely slow. It's strange because this is quite a short one. Maybe it's very unconventional. Okay, let's go for the last one. Oh, not the last one. Oh, yeah, okay, we have maybe more. Okay, the next one. And while these are running here, yeah, so we have a bit more information here. Oh yeah, I know, this is the reason why I wanted to show this to you. So the first thing is that yes, there is an annotated lipoprotein, but we don't really know anything about this. Again, if you check this protein, there is no more information about it. But there is also something here you can see, the DUF1433 domain containing protein. So you may be happy to see, oh, there is a known domain inside this protein. And actually this DUF is an identifier in the PFAM. So if we search this, but of course, before you search the DUF, after your second search for a DUF, you will realize that this is not much better for you because DUF is actually means domain of unknown function. So this was DUF1433. You can just search the name in PFAM, and it's really a PFAM domain category. There is a structure actually about this. It's NMR structure, but we have no idea what's the function of this. There is one thing you can do here. Maybe that will give you a bit more idea on the function of this protein. So you can check the domain organization tab on the PFAM. So even if you don't know anything about the function of this domain, if we have something here, but we don't, then you would be able to see that, okay, this domain is can be seen together with a domain X or domain Y or domain Z. So of course, what we see here is that only this is our DUF domain, and it's sometimes there are two and sometimes there are three. And I think what we have here is just probably the Phoebus, I think, is a signal, or it's a transmembrane region predictor. So there are actually quite a lot of clues now that the protein is transmembrane, but nothing else. If we go back to the thyroidoxin domain, just because I try to show you something. Okay, this one, and we maybe have a bit more about, oh yeah, so this is how it should look like. So even if you don't know anything about your green thing, then maybe you will find other proteins where this green thing is together with something that is known, and you can derive some information from that as well. So probably there are no, random proteins in prokaryotes that when this domain is just randomly put somewhere together with another domain, probably if you find something that you know already the function of, then you can guess some kind of category at least of those proteins. Where are these proteins are sitting in the protein functional space, and you can also find out a bit more information about them by seeing, even if inside your protein there is no fed binding free domain like in this one, but as you can see, the same domain is sometimes together with that one. Of course, we didn't see anything like that for the DUF domain. And if I go back to the next one, I think we can go for the next. This is protein 4. Oh, that was a bad idea to copy it here. Sorry. It's much easier to remove all the spaces here. And it is almost done. Okay, this is a much bigger one. I'm copying it for you. It is there. And I think even on this one, the only thing what we know, oh no, up here, the only thing what we know, it is a conserved hypothetical protein, so nothing else. Again, it's not really informative. Let's try to run the IUPRED first, because it's very fast. As you can see, we have a nice coverage of a full domain here, which is called PyRedox2. So you can just click on the PFAM domain, and you can already see that it's the pyrinucleotide disulfide oxyreductase. Again, PYR redox 2. So even if you don't see too many information here, there is some information, of course. It's not, maybe not exactly what you want to see, but again, if you find that one, which was the same name, but without the 2, the PyRedox, there isn't much more, but there are some literature here, not really too much. No. Yeah, so at least we have some information about it. I remember that I actually found quite a lot about this domain. So if you go after the domain name, and if you go after the similar proteins, then you can find out quite a lot. And it's actually, yeah, it's involved in the nucleotide biosynthesis, or yeah, I think it's in the biosynthesis as well. I don't know if we have any other domains. Oh yeah, so here we have quite a lot of different domain organizations, but be very careful with this one, because here there are a lot of mixture of bacteria and potential eukaryotic species as well. I think mostly here what I see is much more bacteria. So again, what you can see here is that other proteins with exactly the same domain. So again, I think you can have quite a good idea about the function of the protein. If we put the same thing into HMR, which is continuously running on the previous protein. Interestingly, it is called a bit differently here, but I think it's the same domain, PF. Let's see what it found. What is happening if I just click on it? Oh no, it's actually different. It's very similar in function, because it's also net pH dependent. But do we have NAD in prokaryotes? NAD age? I'm not sure. Ibra, do you think we have or not?
I think so, yeah.
Yeah, we have. Okay.
Yeah.
Then should we say that this protein is maybe involved in the energy homeostasis? Not this one, of course.
Yeah, the other one.
Yeah, I think this is not a bad prediction, but again, I'm pretty sure that you can find a bit more information about these if you look at these two domains and try to find out
Yeah.
a bit more about in these publications. So I think with this one we are not bad with the information we were able to extract. Just thinking if we yeah, there are different PFM matches here actually. So the same PFM is also here. And it's actually not bad here. If you see something with a plus, that means that it's a very good, very well matching hit. Right. I don't think we can see too much more here. Not really. It's a globular protein that we already know, so it will be below the line. Just to tell you one more thing about the blue line as well. So the blue line is actually another prediction. So if you have a disordered region, a region which is usually flexible and extended in terms of structure, these regions are extremely good in protein-protein interaction. So for example, if you think about scaffold proteins, let's speak about the ribosome, for example. That's a very good example. Within the ribosome, you have, of course, the ribosomal RNA, but you also have quite a lot of protein parts within the ribosome. And there are a few special proteins, even in prokaryotes, but also in eukaryotic ribosome, that are going through the whole ribosome and act as almost like a glue. So they are responsible for fixing all the other globular or much more sphere-shaped proteins to each other. And of course, if you just express these proteins alone, they don't have any structure. They are not really hydrophobic. They are very often quite hydrophilic and charged. But in the same time, you can very often see very small hydrophobic parts within the hydrophilic part. Let me just show you a very good example. And this will be one of the most well-known protein, which is the P53. Of course, it's not a bacterial protein, but a eukaryotic, especially if the UniProt is working for us, and it's working. So this is P53, which is one of the tumor suppressors in our genome, and one of the most important protein in terms of cancer biology. And if you copy the sequence of the P53 and put it in IUPRED, and then this is what you see. And the reason why I want to show this, because sometimes you can see this in prokaryotic proteins as well, or if you're interested in eukaryotic proteins, that can be very interesting as well to see this. So the P53 has basically three different parts in its sequence, as you can see. Also, if you just look at the red line here, you can see a part in the N-terminal, which is well above the 0.5 line. You can see a part in the middle, which is below, and then you can see a part again on the C-terminal part of the protein, which is again above the line, almost fully, but not completely. We already know, of course, that this middle part of the domain, which is also called P53 domain, two things are involved in this part, the DNA binding region. So, of course, this is a transcriptional regulator protein, so it has to bind to the DNA. And also, there is a tetramerization domain somewhere here, because this protein is functioning as a homotetramer. And we know that these two parts are are well structured. They are both globular, so we know their crystal structure as well. But we also know that the regions here and here are never going to the crystal. So they are not forming one well-defined structure. And actually, the two things, what you have here, these are, both of them are transactivation. They are called transactivation domains. So they are actually docking regions for other proteins. So they are extended peptide sequences with a lot of small short motifs that are serving as docking stations for other proteins. And actually what you see here with the blue line are the potential docking short motifs. If you remember our eukaryotic linear motif database from last week, this is what you have to imagine here: a short linear motif, which is usually identified by a few amino acid constellation, and sometimes it's just 5 or 10 amino acids, and they are serving as a docking position for protein X, Y, and Z. So the protein, this p53, first using the tetramerization domain, going together and form a tetramer, and then binds to the DNA with the DNA binding domain, and then serve as a docking station for other proteins that are also involved in the transcriptional regulation to localize there where the p53 is binding the DNA. Okay, so if you see something like this, a very high red and very high peaks of blue, then you have to think about a protein which is probably functioning in protein-protein interaction. So, of course, for example, the intercellular signal transduction is way much more simple in prokaryotes than in eukaryotes. There are much less signal pathways. Even the transcription regulation is very different from the eukaryotic one. But even in prokaryotes, there are a lot of protein-protein interactions, protein modifications, or just simply dimers that are forming between two different proteins. So there are many cases when you can see very important protein-protein interactions within prokaryotic cells as well. And this is one way to find out if you have something like that, which is again an extra information about the function of that specific protein. So where we are at this protein. Okay, so that was protein 4, which is now, I think, we are a bit closer. Yeah, it was this one. Let's go for the next one. Again, a small protein, so I don't think we will have too much information about this. It's actually, again, conserved, so maybe there will be something about it. I think it will be a quite hydrophobic again. Looking at the sequence. Oops, sorry, that this somehow went into 2 lines. It's strange because it's not in 2 lines for me. It is the same. Oh, I know, because I just highlighted but not copied, sorry. Ah, even worse. Okay, now you have the sequence without any interruption. Okay, this is not extremely hydrophobic, so probably this is not a transmembrane protein. Let's try to find out a bit more with the HMR to see if we have any. If we didn't have any PFM domains. Oh, we have a PFM domain here. Okay. Oh, again, something which won't cheer up too much. Won't cheer us up too much because, again, as you can see, it's a DUF. Let's click on it. So it's, again, a domain of unknown function. The only thing what we can do is try to find out if there is anything else with this domain organization. Nothing. But actually, there is an uncharacterized protein in a Bacillus species. But again, this is not really something which will help give us much more help. I think it is the same here. So again, we have the same PFM domain of unknown function, the DUF2922. So I think, yeah, you can see here again all those similarities that can be found. They are all uncharacterized proteins. So again, I think this is not something where we can help too much to Ibrahim. It is even not a transmembrane protein. So to be honest, I don't really know what to do after this. What we can do maybe is one more thing. Just try to the emboss web info. So go back to the search. Just go back one, and maybe try to find out if there is any special charge distribution or hydrophobic residue distribution within the protein. It won't give us any functional information, but maybe if we know a bit more about the structure, if something looks strange, but nothing looks extremely strange. Charge residues are equally distributed. Polar hydrophobic residues are, again, very similarly distributed. So again, this is something I think we will not find out today. I think we have about 50% of success rate, which is not bad. Not bad actually. It's much better than the Proca, but it's not perfect. So I think we only have one last protein left. Let's try to find out the function of this one. So this will be protein 6. And it is there if you want to do the same thing with me. Also, I will put it into iPad. As far as I see, there was nothing came up. Oh, so this is not that hydrophobic. Maybe this is one of the very rare, a little bit more disordered proteins. There is no PFM domain, unluckily, here. Nothing really here as well. It also says that there is some disorder here. Oh yeah, I remember this one. And it's actually a very good hit in terms of disorder protein. So this is a phage protein, Ibrahim. I don't really know if that means anything. I don't know how often do you see phage proteins that are essential for a bacteria? Can you imagine that it's actually a protein that is probably involved in the assembly of the phage itself, and that is why here we have disorder actually in the protein. It's the same as I told you with the ribosome. So if you think about the phage head is, again, it's a huge complex of many different proteins, and you have to glue them together. And if you have to glue something together, you need this order. So what do you think, Ibrahim?
Well, it's possible because this is like it's a hit from the whole blood assay. So if it turned out to be, because I think Marta and one of our collaborators in China, they already did some bioinformatics analysis saying that some prophage or phage are sort of like only seen in strains of animal lineage.
Oh,
so
that means that there is a.
So it's potentially is unique to this like I don't know actually. So
okay.
I think it's yeah, it could be interesting. I need to look into this closely. Yeah.
Okay.
Yeah.
So at least you know a bit more about it.
Yeah, if it is something to do with different host, that would be very interesting. That's actually we are looking for as well. So
what I would really look into, so especially if you open this, and especially if you open, I think this is actually an alignment with another protein. So there are many things I would like to see. So if this one is a functioning phage in anywhere, so if you can find out a functioning phage, maybe it's not a functioning anymore in your Step 4 use, but if it's a functioning phage somewhere else, then the differences in nucleotide sequence are, I think, very important. So if you see that in your Step Aureus, for example, the GC content or the codon the codon usage is much more typical to the Staph aureus than to the original phage, for example. So here you can also look at the nucleotide sequence of the original phage if you can find somewhere else. That means that the phage is there for probably a long time, and it is not just integrated, but it is also like maintained, you know, in the way that the Staph somehow preserved that gene, and even it started changing to be more compatible with the Staph aureus. But also, I think the protein differences are extremely important, because what I can imagine that if you want to utilize a protein sequence which has a characteristics to interact with other proteins, for example, but the evolution actually introduces some novel function, then maybe this is what you are looking for. So if you can see here, there are small patches, not the single amino acid changes. They are not extremely important, probably, even if this one actually is a huge change, because this is a tyrosine and this is an aspartate, so it's a huge hydrophobic residue change to a negatively charged residue. But if you see patches like this, and especially if you can find more than just one sequence and do a multiple alignment, and you can see that something in your protein is unique compared to the homologs, that's something where I can imagine that you have a kind of carrier protein that was integrated to the genome, and somehow a novel function was introduced into the protein with maybe just a small piece, just a small new linear motif, for example. And actually, I really like the sequence of this one as well. So this is your, this is your query. Yeah. So it's extremely hydrophilic, also negatively charged. If you see a concentration of negative charge in one place, that's always very interesting. E, DD, E here as well. These are all negatively charged residues. I don't know if you can color it here. I'm not sure. So this is what I would do for going forward. Try to find as much homologs as you can. It's not very hard because you have all of them here.
Okay.
Just download these sequences. I don't know how long it is. This is not extremely homolog, but
That's something.
yeah, it's not bad.
Yeah, thinking about that. So what I did with that, because yeah, we're obviously interested in protein and nucleotides. So if you have a gene of interest, what I did, I built a phylogeny just to try to see if there is a structure by host on protein level or on nucleotide level. Mostly nucleotide, but then I looked also at protein, you know, synonymous and non-synonymous mutations. But it didn't seem to, you know, like correspond to population structure. So it happens multiple times. Not in this specific gene, but one of them. So that's what I started thinking about with that.
Yeah, absolutely.
So you'd say about how many mutation within the region, or how many amino acid change would you say is potentially leading to change of function? If it's one, it's probably not a big deal, right? Tend to be.
There is also a known protein like this, but it's much shorter than yours. So it is also, maybe there is a probability that this protein can be truncated somewhere. So maybe it's also worth to try just to make another search with a truncated version of this one. I think this is the same protein, but only the beginning, the first 100 amino acids. Maybe you will see this much more in other organisms. If you look at this region here, so it's clearly the first part is the part which is responsible for the protein-protein interaction part, because there are these disordered patches there. So I can imagine that you will find the same kind of sequence somewhere else, but in a shorter form. I would try to go with the first 100 amino acids. Again, you have to look through all these hits, how they are aligning. There will be some other staph aureus as well. You will quickly learn that if you see stau, that's what you want to see. That's the Staphylococcus aureus abbreviation for the uniprot, or STAA as well. Here again, yeah. So there is one annotated region in this strain as well, but only with the beginning of this. Maybe I'm just thinking, because you did a Tradis on this one, maybe it also worth to go back to your Tradis and check where if you have any insertion events in this gene, but maybe it's not within the whole gene. You know, if you cannot
be
see,
under the threshold, yeah.
Yeah, if you cannot see not just the threshold, but I think you can map, or if you don't know how, I think I can help you.
And to see which part is
to map all your Tradis insertions on the gene, and do it for all of your environments actually, and see if there is some kind of preference of insertions or some kind of avoidance. So you know, if this is 100 amino acids, that means it is 300 nucleotides. And you, as far as I remember, you have quite a dense library. So like every 10, or on average every 10 or 15 nucleotides, you had an insertion. So I think it's worth to look for if you see a 300 nucleotide intact, but there are other insertions in the 3' part of the gene, that would give you an idea on the importance of the first part, maybe only the first part of the gene. Right, this was a bit shorter than I expected, but I think we that's for now. But if you have any questions or comments now for this, I think these are the most important websites you have to be aware of if you are doing any kind of structural approach to the functional word. There are, of course, servers, meta servers. There are always papers about new methods that are doing the same thing for you in one server. I also tried to find one, but the very first one I found was a hyper super paper from 2019, so it's only 1 year old, and it promises a perfect functional ontology for you. And when I ran the server, it gave me a server error. So the reason why I showed you these links, because these are all, you know, very well maintained programs. The NCBI BLAST will always work. The EBI AGMR will always work. The UniProt is always up and working. And actually the PFAM is the same. Sometimes the PFAM slows down, but usually it is quite reliable. And I also know that the IOPRED is a very reliable one, because just simply because I know the guys who made this server. Right. Any questions or comments?